everyone. Today we are going to study about the maturation of B lymphocytes. Well, first we must know that what are these B lymphocytes? These B lymphocytes are a type of granulocytes which are present in the body and they generate an immune response during an antigen invasion. Okay, and so now we must first know that from where these at what place these B lymphocytes are actually maturing and originating. Okay, so the origin of B lymphocytes lies in the bone marrow. Okay, in the bone marrow, these B cells have their origin from the hematopoietic stem cells. Okay, these hematopoietic stem cells, they have the capability to form any kind of blood cells, like can be a B lymphocyte or can be a T lymphocytes or any other granulocytes or granulocyte. Okay, so the first stage, or you can say the first distinguishable B cell in the B cell lineage is the pro-B cell stage. Well, it is just the nucleus and the cytoplasm and no other membrane-bound cell receptors are present in this pro-B cell stage. Okay, so now this pro-B cell stage is converted into the pre-B cell stage where we have the heavy chain of the immunoglobulin being incorporated into the cell membrane of pre-B cell. Along with this, you have two transmembrane molecules, Ig alpha and Ig beta. Now, how does these heavy chain as well as these transmembrane molecules are being formed? The answer is simple. There are four kind of genes which are responsible for this formation. Okay, these genes are V, D, J, and C. Okay, when these genes are productive, you have the formation of the heavy chain of immunoglobulin. Okay, and so this is what is happening when we are moving from pro-B cell to the pre-B cell stage. Basically, the rearrangement of V, D, J, and C is taking place inside the nucleus, resulting into the formation of the heavy chain of immunoglobulin. Okay, now as we move from pre-B cell stage to the immature B cell, you have the formation of the two light chains of this immunoglobulin. That is, you have final and immunoglobulin M being formed on the surface of this B lymphocyte. Okay, and this stage is known as immature. Why we are calling this stage as immature B cell? Basically, this immature B cell, according to studies, have been found are not capable to form the antibodies. Okay, now this immature B cell will then further be transformed into the mature B cell by the formation of the other membrane-bound receptors or B cell receptors as well as the membrane-bound immunoglobulin IgD. Okay, so this is what you see in the next stage, that is mature B cell. We have the formation of the other cell receptors, that is B7 or MHC molecule class second, as well as this Ig alpha and the Ig beta being present also, and with an immunoglobulin M and an immunoglobulin D. Okay, along with these receptors, which I have drawn in the diagram, there are other receptors as well. Okay, so basically on a mature B cell, you have many kinds of B cell receptors present, and this mature B cell will remain inactive till an antigen is not encountered either with an antibody or is not presented to the MHC class second molecule. This mature B cell will then be transported to the secondary lymphoid organs present in the body. Well, the secondary lymphoid organs, for example, are Peyer's patches or Malt, or you can say the lymph nodes, basically. Okay, so when these are transported, these mature B cells are transported into the secondary lymphoid organs. They remain in this mature B cell stage unless and until an antigen is being presented to them by the antigen presenting cells, for example, macrophages. So, for example, if an antigen has just been presented to these mature B cell, now these mature B cell will start division and form clones of themselves. Okay, and multiple copies of this mature B cell will be formed. Okay, but basically these all clones are not selected. Only those clones are selected which have the capability to form that antibody which is capable to interact with the antigen which has just encountered with this mature B cell or with an antigen presenting cell. Okay, so now out of these clones, only certain mature B cell clones will be selected, and these selected clones, they further activated, they further undergo division and formed B memory cell and the plasma cells, and these plasma cells are actually the end stage of B lymphocyte maturation. Okay, and now these plasma cells, they are capable of synthesizing any kind of antibody like IgD, IgE, IgA, IgM etc. Okay, and so this is how a B lymphocyte is mature, going from a pro-B cell stage, then the pre-B cell stage, then immature B cell, a mature B cell, and finally division into a plasma cell and the memory B cell stage. I hope this was helpful. Thank you. Keep watching.